Bismillahirrahmanirrahim. So today we are going to cover the last lecture about fly development in terms of segmentation. We will keep coming back to fly when we will talk about fertilization, when we will talk about organogenesis, differentiation, etc. But as a model system, to answer a question about how segmented body plan is established or how pattern formation takes place during early development. So we are going to talk about the master regulators of development, the genes called homeotic genes. So let me share my screen. So as usual, let me recap our last lecture where we talked about segmentation genes and their role in specification of segment identity. So these were last few slides we covered. And in terms of compartmental boundaries, what we also saw that we saw that the compartmental boundaries which are established early on during development, they are preserved throughout the development all the way to adulthood as well. And even in the adult abdomen, each segment is subdivided into a number of types, like which are given here: A1, A2, A3, A4, A5, and A6. There are at least 5 sub-compartments, or 5 regions within anterior compartment and 3 within posterior compartment. And the patterns of bristles, pigmentation, etc., they are linked to the specific gene expression pattern in each segment, and all this is established early on, which is then maintained during later development. We also talked about in previous lecture about planar cell polarity, where each cell in the body has a polarity. Each cell has anterior and posterior polarity. For example, if you look at here a cell bristle which emerges from the abdomen, it points towards posterior side. So it means this cell has a specific anterior and posterior polarity, and that phenomenon is called planar cell polarity. And if there is disturbance in the compartmental boundary, which is this red one, the cells lose their polarity. Cells, they simply get disoriented, and a symmetrical development of pattern of body is completely messed up. And you see, due to disturbance of this compartmental boundary, you lose the not only planar cell polarity, but the whole pattern is disturbed. When we talked about segmentation genes, we specifically talked about denticle belts in the abdomen of larva. And we learned that anterior region of each compartment, so anterior region has unique denticle belts, and the posterior region of the segment, it's naked. And each segment has unique pattern. And this pattern, we learned that this pattern is due to winglets and hedgehog together with the engraved. And how we know this? Because through genetics, once we had wingless mutant, the pattern was disturbed. We got mirror image of, so this image was, this denticle belt was reproduced in the naked region as well, and this one here. And when we had hedgehog mutant, we saw that phenotype was same. So that provided us clue that, hedgehog and wingless work together. And in last lecture, we did learn that at the compartmental boundary, anterior, posterior compartmental boundary, the cells which are lying at the boundary, they are actually communicating with each other. Alhamdulillah. So you have, for example, engrailed here, which activates hedgehog. Hedgehog is a paracrine factor which diffuses towards posterior side. It activates through hedgehog receptor, hedgehog signaling. You have activation of wingless, which then comes back, and this compartmental boundary is maintained. Now we are going to talk about homeotic genes, which play a role as master regulators of development, and they are they are so crucial in development that all the work which is laid down by, and this is the cascade of gene expression, what we have covered so far. So with the maternal genes, immediately after fertilization, we have Bichard and caudal. This is the Bichard staining on this side. This is the caudal staining, the green one. And then we talked about gap genes. We have, you see, NERPs, and then Krupel. This is also NERPs. Gap genes disappear, they are transient as well, and they activate the pair rule genes. So this is even-skipped, and Fushitarazu. And then we get the segmentation genes. Their staining is not here. We just saw segmentation genes and wingless, hedgehog, etc. Now the point is, they are transient. They are transient. They are transient. These are cell signaling factors. By this time, we already have the specification of the cell fates. Fates of different cell types, they are specified already here. We just saw this one. This is late developmental stage, but at embryonic development, we already have cells whose fates are specified. They have committed to specific cell lineages, and after mitosis, these cells are, their fates are maintained. However, once the segmentation genes established, someone has to, lock the cellular identity. Someone has to play a fundamental role to ensure that, this passing on baton to the next class of genes finally comes to an end, and the segment identities are fully established, and then they start maintaining that. That's where the role of homeotic genes come. Homeotic genes are the ones which give specific identity to different body segments during embryonic development. And these homeotic genes, they are transcription factors. They are, their expression is established based on this cascade of gene expression through cell signaling by segmentation genes in a segment-specific manner. If you have anterior to posterior embryo, if I draw here, so you will have very, very specific expression pattern of homeotic genes, and soon we'll see what these homeotic genes are. And their characteristic expression in different segments needs to be maintained forever through the expression, forever throughout the development, all the way till adulthood. And that expression pattern is maintained by a group of genes which are called polycomb group and trithorax group protein. This is also the subject of research in my lab. So since, let's say a homeotic gene is on here and off in these segments, so this class of genes, polycomb group, is going to maintain the homeotic gene in the off state here, and trithorax group antagonize repression or silencing by polycomb group, and they maintain genes in the on state here. We'll talk in great detail about this. These are chromatin binding proteins, but today's lecture is about homeotic genes. What is role of polycomb trithorax? That once they come into action through mitosis, they then maintain the expression pattern. So in all the cells where trithorax group has maintained gene expression of a specific homeotic genes on, same gene is off by polycomb group. So these are chromatin binding proteins. These are, they bind to chromatin of homeotic genes and maintain in the on or off state. And the phenomenon which we during development refer to is called cellular memory, because once a gene is on in the cells, all the future progenitors through mitosis, they are going to maintain the genes in the on state. Yeah, this is a cellular memory. So let's say you have specific genes, homeotic genes off, and one of the key homeotic genes on in another cell lineage. These two genes are off and this one is on. Through mitosis, you can see the progenitor cells of this lineage will maintain these two in the off state and this one in the on state. And this is the function. So here, silencing or off state is maintained by polycomb group and on state is maintained by trithorax group. At different cell lineage, so different genes off, a different gene is on, and through mitosis, they are inherited in the off and on states. And this function is done by polycomb and trithorax. I told you, so trithorax maintains genes in the on state and polycomb maintains genes in the off state. They bind to the chromatin regions and then maintain them. So what are the homeotic genes? Homeotic genes are the ones, as I told you, which control the identity of specific segments. Segment identity, which was established early during development, which kicks off through action of maternal genes, followed by gap genes, payroll genes, segmentation genes. Eventually, this segment identity is established, or the specification of segment identity is established and maintained by expression of homeotic genes. How did we discover homeotic genes? So, and what are the homeotic genes? There are two clusters of genes in fruit flies. And these two clusters of genes are, one is referred to as antennapedia complex. The other is called bithorax complex. This is on the chromosome 3 in fruit flies. Okay. Now, in late 70s and early 80s, in Walter Gering's lab, they discovered these genes. And what they discovered, that all these homeotic genes, they have their proteins, they have specific DNA binding domain, and they name this domain, this conserved sequence of amino acids, as homeobox domain, because they recognize specific sequence in their own promoters. Okay, their promoters have the recognition site, and the protein has the specific domain called homeobox domain. Now, this hash line is indicating a long distance on the same chromosome. And in the Antennapedia complex, the genes are called antennapedia, sex chromosomes reduced, deformed, proboscipedia, and labial. In the bithorax complex, the genes are called UBX, ultrabithorax, abdominal A, abdominal B. And this is the way how these genes are organized. So if this is the 5' side of the chromosome and this is the 3' side of the DNA, so this is how these genes are organized. What is interesting that this cluster of genes in fruit flies, the way they are organized and the way they are expressed in fruit fly embryo. I try to draw embryo. Labial is expressed in the anterior most region. Proboscipedia is then expressed. Then is deformed is expressed. You go from anterior to posterior of the embryo and you will find abdominal B is expressed in the last 2-3 segments of fly abdomen. It is then followed by expression of ABDA, which is then followed by expression of UBX. So this expression pattern from anterior to posterior, I mean, it's very surprising because each cell in the body here, each cell contains this chromosome. But they are very selectively, different cells are in different segments of body expressing just labial, then proboscipedia, then antennapedia, etc. This expression pattern, as well as the organization of the genes, homeotic genes, is 100% conserved if I try to draw the mouse embryo here. Okay, if this is mouse embryo, in the mouse chromosomes, these genes are not only conserved in the same order, but their expression is also conserved just like we have seen expression here. So we call this phenomenon conservation, and conservation not only in terms of genome organization, but also the expression from anterior to posterior of the body. We call this phenomena collinearity. It's like a linear, arrangement. It's not due to chromosome lying this way, that chromosome is lying. Don't think like this. Chromosome is present in each cell here. But it's just precision engineering, I call, or developmental plan, developmental toolkit, which establishes this gene expression pattern. And then each of the homeotic genes, for example, labial, gives identity to tissues and organs here. UBX gives identity to tissues and organs here. So we are going to just pick up one example here in the thoracic segment. So this is the T2, thoracic segment 2, thoracic segment 3. The Drosophila wing is attached to thoracic segment 2. This is the anterior compartment. This is the posterior compartment of the wing. In the thoracic segment 3, the organ, which is a bulb-like small structure, which is called halter. Halter is involved in, maneuvering the flight of fly, maintaining the balance of fruit fly during the flight. Now, if we have mutation in bithorax complex, what we discovered that the halter, it is converted into a wing. So essentially what has happened, wing is normally attached to T2. So what has happened, T3 is getting converted into T2. This is transformation of one body segment to another. And this phenotype is classically called bithorax phenotype. Look, this fly has two thorax. This is the one, this is the other. And now, due to transformation of T3 into T2, whole of halter is converted into wing and thorax is duplicated. This is called classical bithorax phenotype. These flies are unfit. They cannot survive longer because they cannot mate, they cannot fly. So they are very difficult to maintain in the lab. But this is, look, you have now two wings on each side, so two pairs of wings, which is basically telling one of the body segment is transformed into another, and this phenomenon is called homeosis. And from this word homeosis, the word homeotic transformation was discovered, or the name of the genes was coined as homeotic genes. Okay. Now, let's look at some of the examples which are linked to the bithorax complex, UBX, ABDA, and ABDB. If we take antibody against different genes in homeotic cluster in bithorax complex, for example, anti-UBX, anti-ABDA, anti-ABD, ABDB, and we try to see the expression pattern of these genes in today's time, in 21st century. You will see a picture like this is immunostaining. You know, I've used the antibodies to stain these embryos. One of my colleagues during my postdoc lab, Nara Lee, he prepared these images just for learning purpose. This was a just purely learning experiment. This was not part of the research, to see through our own hands how they look like. If we use UBX antibody, abdominal B, and ABDA antibody, we will see a very characteristic expression pattern. However, how through genetics we can understand the bithorax complex? So we'll come back to this antibody staining and how in the present day we can solve this question. But let's first look at classical genetics approach, how they learned this, how they understood all this. That bithorax complex or genes within the bithorax complex, they are giving identity to specific segments. So what they saw that when we have intact ABD, ABDB, and UBX, this is the wild type bithorax cluster. You know, this is how the parasegments are present in the larva, and these are the segments. T1, T2, T3. Head is usually fused, so we cannot point out head here. A1, A2, A3, A4, A5, A6, A7, A8, the abdominal segments and thoracic segments. If they have delta, which means you have deletion of bithorax complex, all three genes are deleted. What was the phenotype? Goodness. Look, 3, 4, 4, 4, 4, 4, 4, 4, and just 40. T1, 2, 2, 2, T2, 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 everything. So the parasegment 4 or thoracic segment 2 has taken up identity. All these segments, all the posterior parasegments, which was parasegment 5, 6, 7, 8, 9, they all are transformed into parasegment 4 or T2. Isn't it mind-blowing? Doesn't it, blow you away? What is happening? It indicates there is something happening here. There is, in wild type case, between 4 and 5, 5 and 6, 6 and 7, there is some sort of antagonism going on. There is some sort of, if that buffer is gone, it flows, it floods these segments and take up their identity. Homeosis has taken place. Okay, we have this now. ABD B is absent here. ABD A is absent. UBX is absent. And morphological phenotype, which is definitely not going to be viable. Now, said let's put back UBX. Let's bring in this mutant UBX alone. What has happened? 5 and 6 came back, but now 7, 8, 9, 10, 11, 12, 13, they all are transformed into 6, which means there is an antagonism. There is a buffer here between 5 and 6. And if that buffer is gone, this is flooding into all this and transforming parasegment 7 all the way up to 13 into parasegment 6. Okay, now in addition to UBX, now let's bring back ABDA. You have restored 3, 4, 5, 6, 7, 8, 9, 9, 9, 9, 9, and 10. So what are you concluding? The conclusion is that genes within the bithorax complex, they are restricting each other. They are having kind of boundary within their expression patterns. And what today we know that, so if you bring back now ABDB, B, you see, and here, what is missing? You have ABDB and ABDA is there, but UBX is missing. Look, here, when UBX is missing, what has happened? 3, 4, 4, 4, 7, 8, 9, 10, 11, 12, 13, 14. So the domains of ABDB and ABDA and ABDB expression, they have specified, they have maintained identity of abdominal segments of their own domains, but the domain of UBX is taken up by parasegment 4. And I told you, so from all the anterior segments up to parasegment 4, we have expression of labial, proboscipedia, deformed, sex-reduced, and antennapedia. UBX starts from parasegment 5, parasegment 6. Parasegment 7, 8, 9 is ABDA. And so what is happening? The genes are inhibiting. The most posterior gene is ABDB. It is inhibiting expression of ABDA. So in parasegments 14, 13, 12, ABDB is expressed and it suppresses ABDA. Similarly, ABDA expresses in 12, 11, 9, 7, etc., and it suppresses UBX. UBX expresses in 5, 6, and suppresses antennapedia. Now, once UBX is gone, antennapedia spreads. It takes over the segments whose identity is maintained by or who are specified by UBX. Look here. This is 5 and 6 is the expression of UBX, which maintains here. UBX is expressed, you see here, weakly in other segments as well, but the identity of these segments is maintained by ABDA then, and then these segments by ABDB. Is it clear so far? Ji Fariha?
Okay, so I have a question. So is that because what you just said that ABDB is so each homeotic gene or bithorax is gene or is basically suppressing the other one. Like ABDB is suppressing A and then it's in turn suppressing the UBX. So is that because like we see here that the parasegment 14, it's still there when all three are absent in the second image. So is it because the parasegment 14 is because of the expression of the ABDB and it's not because the ABDA is being suppressed by B, something like that?
So we see 14 there even if we remove the whole bithorax complex. 14 is there, A9 is there. So it could be a different developmental gene which is maintaining most posterior segment, because it is there even in the absence of ABDB. Did I answer your question?
Yes, sir.
So now, yeah, so what we have covered is that UBX we knew from wild type 5 to 12, ABDA 7 to 13, and ABDB 10 to 14. If you bring in UBX and ABDA, which was this one, you restore 7, 8, 9, and 9 spreads and transforms the ABDA transforms identity of all the most posterior abdominal segments. Now, who maintains this? Once homeotic genes, they start expressing and give specified identity to these segments. So this is what I told you, polycomb group and trithorax group. They are the last in this cascade of gene expression. They then maintain the cell identity. I explained you how they maintain. So UBX will be maintained on here in these segments by the trithorax group action, and it will be maintained in the off state wherever UBX needs to be off. It will be maintained in the off state by polycomb group, and then through mitosis, each successive cell division, these expression patterns are maintained. They are faithfully inherited to future progenitors of mitosis, and the cell identities are maintained. So this is what we have covered. So we have the gap genes, payroll genes, etc. Then we come up with the selector genes of the homeotic complex. We talked about bithorax complex. We talked about antennapedia complex, their collinearity, their expression pattern. The antennapedia complex is labial, proboscipedia, deform, sex chrome reduced, etc. I told you they specify the identity in the head and first thoracic segment up to parasegment 4. And then the bithorax complexes, UBX, ABD, ABDB, they specify identity in second and third thoracic segment, which are basically all the way from parasegment 5 to parasegment 14, or T2, T3, all the way to A8. Okay. Now, what is time now?
10:40, sir.
10:40. Let's take 10 minutes break, and then we continue with part of recitation, because it's good to cover this lecture, what we are learning, and do a do a question. Will using different tools, like for example antibody, we talked about in situ hybridization, we talked about making transgenic flies, etc. So after 10 minutes break, we'll come back and do a practice question which will help you understand how we use different cell and molecular biological tools to understand development. For example, we will take this example. We are with UBX. We will take some UBX example and understand how we apply these tools. Okay? Is there a question? So if there is no question, let's take 10 minutes break, and we gather 10:52. On my computer, it's 10:42, so I'll come back after 10 minutes.